Welcome everybody to our Diversity in STEM series. I'm Dr. Morano, and I have the great honor of introducing today's Diversity in STEM Scholar speaker. So today's speaker is Dr. Christopher Bolden. He is an NIGMS Trauma Postdoctoral Research Fellow at the University of Texas Health Science Center at Houston, right down the road here in surgery and pediatric surgery. And he's received a BS in biological sciences and biochemistry from the University of Southern Mississippi. And there he studied staphylococcal virulence factors. And then he went on to receive his PhD in interdisciplinary biomedical science with an emphasis in clinical and translational science at the University of Arkansas. And while in graduate school, his research focused on methamphetamine antibody gene therapy. But right now, his research focuses on characterizing a novel platform to investigate trauma brain injury in vitro. And so I think it's going to be a really interesting presentation. For those of you guys who are listening, I just wanted to tell you a little bit about the format of this day's presentation. Dr. Bolden's going to start with a discussion of his story, like how he came to be where he is now. And then he's going to dive into his science and tell us a little bit about his science. His goal is to finish around 12:40-ish. We're going to open it up for some science questions. And then at 12:45, I'm going to ask that all the faculty leave. And Dr. Bolden's going to be on this webinar from 12 all the way to 1:15. And he's going to field questions. Students can ask any questions they want. And in order to ask those questions, you're going to just type them in the Q&A. Between 12:45 and 1:15, he's just going to answer as many as he can to try to inspire you all in your future career paths. I think that's it. So I think that's all the basics. So Dr. Bolden, why don't you try to do share screen? Let's see if that's working.
Okay. Is it sharing?
Yeah, that looks awesome. And I know you have a lot of good stuff to tell us, so I'm going to just have you take it away.
All right. So today I'm going to tell you about my career trajectory into biomedical research. So I was born and raised in Natchez, Mississippi. Natchez, Mississippi is the oldest city on the Mississippi River. And while I was there, I attended Morgantown Elementary School. At Morgantown, I participated in science early on. So we had an opportunity to do a lot of early career science in the Upper Bound program at Alcorn. Alcorn State University is a 4-year HBCU that is just 30 minutes up the road. So Natchez has a very strong connection with them. And they try to get students like me involved as early as possible. While I was at Morgantown, I really got involved in school and statewide science fairs. So during my trajectory at this point, I was primarily looking at ecology and journal biology. So I started doing volunteer work with the Mississippi Wildlife and Fisheries. So I was really interested in the different ecosystems of Natchez. We have the Natchez Trace, which goes directly up through the heartland. It was just an amazing opportunity to get involved in my state. So it was a good chance to get involved in research and science, but also get involved in just the overall betterment of our community. And while I was at Morgantown, I was very involved in choir and band. Interesting fact about being involved in choir is that I had my exact same choir teacher from 2nd grade to high school, and I'm gonna tell you about that as we're going through, and how she still remained a great mentor and now friend after all of those years have passed. So after Morgantown, I went to Robert Lewis Middle School. At Robert Lewis Middle School, I continued to participate in the local and statewide science fairs. I was directly still looking at ecology and the different ecosystems of Natchez. We had different freshwater creeks that go through there, and there was different organisms that were only found in these very interesting habitats. So I was really involved in just understanding how this ecosystem affected and how did the people of Natchez affect this ecosystem. Because at this point, Natchez was starting to slow off from being a major factory run town to be more of tourist a tourist base. So we were getting a lot of influx of people, and we're no longer having as lot of air pollution going on in the city. I continued on with choir. Didn't fall in love with band as much as I thought I was going to fall in love with band during elementary. So now I was moving on to just completely focusing on choir and science. At this time, because of all of my work I had done with the wildlife fisheries, Mississippi Wildlife and Fisheries, I became a Mississippi Delta Science Scholar. And so this gave me the opportunity to do funds and work on community events to increase the number of students in our community or in the Mississippi Delta into science. After finishing at Robert Lewis Middle School, I went to Natchez High. So at Natchez High, I was in the high school, and we had a vocational center. So my my interest started to change a little bit. At this point, being a student at Fallon, after 2 years and passing certification, I started to really get interested into technology. And so I became a computer network technician while I was there. I continued to compete in science fairs though. I really was still interested in more marine biology at this point. So I had looked at freshwater biology and starting to get more interested into marine biology. Being so interested in science and wanting to understand my ecosystem so well, me and my best friend, who you'll see a picture of later on, we found the NHS's Biology and Ecology Club. This is where we got our first full working in a laboratory research experience. During this time, we got really in connect with the University of Southern Mississippi. There was a researcher there named Dr. Ellender, Dr. R.D. Ellender. He was very interested in enterococci and Gulf coastal waters after post-Katrina. We got to go down to Hattiesburg on the weekends, work in his lab, go to visiting like the coastal region of Mississippi, like Biloxi and Ocean Springs and New Orleans, gathering water samples from different water ecosystems, as well as testing our water sources and water plants, and evaluating the presence of enterococci, because there have been a lot of different illnesses associated with enterococci in that area. While still in Natchez High, I was the class president. And class president, I really loved the ability to have a leadership experience. So during this time, we created a district-wide tutoring program. This district-wide tutoring program was because of Mississippi's state test. So Mississippi have state tests in biology and algebra and English and different subjects. And I really wanted to make sure that students were passing these tests successfully. So this became almost a full-time commitment. And this program is still going on to this day. So I worked with the mayor as part of the Mayor Youth Council and as an NHS class president to get more funding. And we really had a strong commitment from the faculty on campus and from Natchez's upper leadership. We were able to receive funding for snacks, for printing off resources, having sites to do these tutoring programs from 4 o'clock to 6 o'clock, and even providing, if necessary, transportation for some students who didn't have a way to get home but were in desperate need of this different material that we were teaching during these tutoring programs. And these tutoring programs were all led by students just like me, senior students who were interested in their own community, making sure that we were succeeding and going on to whatever their postgraduate plans were. After doing all of this work in high school and doing all this work with the different biology ecology club, and doing these different testing, is when I really realized that I might want to actually do biochemistry. So I still love freshwater ecology, but I love the biochemical testing aspects of it. And I also love this leadership and teaching people about STEM, like math and science, but also helping people reach their potential. So I really love this leadership training, and I really wanted to go into biochemistry. So around this time, after working with Southern Miss for such a long time and applying for the different scholarships, I was offered the USM Leadership Scholarship. This scholarship paid for my tuition as well as my room and board. So I had a full scholarship there. And in the biochemistry department, I was also offered their Promising Freshman Scholarship. So at this point, I felt really comfortable about going to college. Even though coming from a smaller rural area, I felt like I was determined and ready to go off. And so I did. I went to college 6 to 7 days after graduating high school. I know that's kind of crazy, but I was super laser focused. I graduated May 21st. I moved into my dorm on May 27th, and school started, I believe, May 29th. And so I instantly got involved into the community at Hattiesburg, and it just continued from there. And just looking at a couple of these pictures, this is our auditorium at University of Southern Mississippi, or just USM as it'll be known in this PowerPoint. But this is my best friend I met during my first day at Southern Miss. We both ended up graduating as microbiology majors. I got really involved in the Lewis Stokes program. And so this is us as a group at our statewide convention. I'm going to talk about some research experiences. So I got to do research just not at Southern Miss, but at different institutions across the United States. And so this is a group of us who started together as RU program at the University of Iowa. And most of us now are either PhDs or MDs, and we're still friends. I was in one of their weddings not too long ago. And this is just a picture of me and some friends at USM outside of the biology building. So let's talk about this trajectory. So I had started to get these early aspects of that a research career might be the focus for me. So now I was time to start college. And so as a freshman, I started as a double major. I was very, very motivated, and I wanted to do biochemistry and computer science. During this time, I joined the laboratory of Dr. Anthony Bell. He is still my mentor to this day. He is who I did my honors thesis project with, and he eventually served as a member of my dissertation advisory committee. So let's talk about my interactions with Dr. Bell. Dr. Bell now is an associate professor at the University of San Diego, but he really brought me in, and this is really what's the focus of my career trajectory, is having these strong mentors that are going to make a big impact in your life. I was a freshman student wandering the hall trying to figure out who my advisor was, and he brought me into his lab. And he was like, we don't always see students like you up here on this floor. So he was interested in me just joining his lab. And I was interested in just trying to figure out who my advisor was. And he was the person that I was looking for. And I started to work with him. In his lab, I learned a lot about the basics of molecular biology, because before I had done some biochemical testing, but it was a lot of field work. This was actually sitting in a basic biochemistry lab and learning the techniques of cloning and purification of DNA. He set the expectations of what I was going to look for in a mentor, and he has been the gold standard of what I need and what I want in a mentor for my academic career. So after this successful first year, some things had changed. I no longer wanted to be a computer science major, but I did want to look into microbiology. I switched because of the interaction I had with one of my TAs during my first year. She talked about the importance of microbiology and how similar it was with biochemistry. And she also just became a great individual mentor. And during this time, as switching to a microbiology major, I was offered a unique experience as a sophomore in undergrad. As a sophomore, I was offered to be a TA for Biology 103. So this is biology for non-majors. And in this capacity, I was teaching 2 laboratory sections of Biology 103 as a sophomore. And it was interesting because I was also a resident assistant at this time. So I had students that were in my floor, or their girlfriend or boyfriends, also students that were older than me. And they were thinking that I was a graduate student, but I was a sophomore teaching them about how biology affects our everyday life. And this is another example of purpose of strong mentorship. So during this time, while I was teaching, I got to really know my microbiology advisor. So she became my advisor during my second year. And so on her door, as you've probably seen outside the office of most of your professors and advisors, there's always some kind of flyer there. And the flyer is either offering some kind of opportunity or different events that are going on campus. And I saw a unique experience. It says Summer Research Experience for Undergraduates, which is RU program. And it was at the University of Iowa. And I talked to her, I was like, is this something I can apply for? She was like, yeah, you're a sophomore now. She said, you can apply for it. She said, I think you'll make a strong candidate. And I was really nervous. So I applied for this opportunity and I got it. I remember I had just finished teaching a laboratory section, and I checked my emails and it said, congratulations, you are now selected to attend the University of Iowa's RU program. And that was actually my first time flying as well, was going for this internship. And during this time, I also got really involved as a member of the Lewis Stokes program. And we reformatted our program to be named IMAGE, which is IMAGE stands for Increasing Minority Access to Graduate Education. And we eventually reformatted this program because we wanted to make sure that we were building as much as an inclusive environment at this institution for students to come in and figure out how they were going to get into graduate school, or even some students that didn't want to go to graduate school, how are they going to get into their professional school. So at Iowa, this is where I met Dr. Patrick Sleiber. I only call him Dr. Patrick Sleiber doing these presentations. Within his first day, he told me, I do not go by Dr. Sleiber unless you're outside of my lab. If you're in my lab, you call me Pat. And Dr. Sleiber worked on a lot of Staphylococcus virulence factors. So I worked with his studies, looking at patient samples and looking at different community associated strains of Staph aureus that were being transmitted locally. And it was a great experience because in his lab, I learned about the fundamentals of molecular and microbiology, and I gained a lot of independent experience. Once I was trained, he felt comfortable in me taking on additional projects during this summer. I would eventually come back to work with him during my Christmas breaks. I went, as soon as I got out of school for 3 weeks, I went back to Iowa during the winter, which a Mississippi boy going to Iowa in the summer is a big difference. And I got to work with him during the summer as well, during the winter as well. So my mentorship kind of continued after this. During my junior year, as a member through Lewis Stokes, I was chosen to go to the University of Belize. And this was a great international experience. I got to stay at a wildlife sanctuary and see the different ecosystem and environments going on there, but I also got to do international research, which is amazing. And this was I was a little nervous of going for this experience because at this point, I was still in the school semester currently going on. So we went during April, and I still had a month and a half of school that I have to also keep up with while I was there. So while I was at the University of Belize, I got to look at the ability of hospital-acquired infections of staph, which was having a major outbreak at that time, as well as looking at some ecology, looking at the lionfish invasion. Right after, literally, as you can see my story, I'm going from one thing to the next with a short intermediate time between. 2 days after returning to Iowa from Belize, I went to the University of Iowa again. So I had been accepted into a different RU program at another institution. And I wrote it on Facebook. So this is a great example of how social media, you don't know who's watching your social media. I wrote on Facebook, I'm so excited. I am going to Wisconsin to do research in microglia. The next day, I got an email from the University of Iowa saying, congratulations on being accepted into this program at Madison, but we would like you to come back to Iowa as an RU mentor. And so I accepted. I got to come directly back to Dr. Sleiber's lab, and it just continued on from there. As an RU mentor, I was really trying to build a great experience for the students that were just their first time in Iowa as RU, as well as completing a complete independent project at this point. So I was an undergrad completing an independent research project in his lab. When I returned back to Southern Miss during my senior year, I worked with Dr. Shu. So I was still working with Dr. Bell, but I thought because I didn't have a lot of courses left, I was still taking classes online into summer while I was doing the different RUs. So I didn't have a lot of classes to take during my senior year. So I thought I could do 2 research projects. And I did. I worked with a research project with Dr. Shu, and he works with mast cells. So Dr. Shu's experiments were looking at how mast cells secreted these different granular substances into their cellular environments, and how did they regulate these different important mediators. In his lab, I served as his lab manager. So at this point, I was learning a lot about what it took to learn budget and finance in undergraduate lab. From his research, we submitted a Center for Undergraduate Research grant. We got funded, and it also helped pay my stipend as working in his lab. So now at the complete culmination of my undergraduate career, I started to think about what would be some things that led to my success. And so this is actually shared at Southern Miss from years ago. And this is because at Southern Miss, we tried to create an environment of each student. So a senior student should take on a freshman student and be their mentor. So I wanted to leave behind just a bunch of advice for students who are trying to get involved in science or just in their undergraduate career. And so the first thing was to study and get to know your professors and advisors. A lot of times we feel that, oh, these professors are so high up that we can't talk to them. but you really got a great opportunity to talk to them during their office hours or emailing them and just talking about what your goals is and how they can help you, because as a professor at an undergrad institution, they are committed to your success. So I really got the chance to just to get to know them personally. I communicated often a lot with them. So I made sure that I was doing really good email etiquette with communicating with these different faculty members. I discovered what my passion was. And so at this point, I discovered that my passion was biomedical science. I liked all aspects of biomedical science, but at this point, I really thought I was going to be a microbiologist. I just developed what a strong work ethic was. So in high school, I felt like I didn't need to study as much as I did in undergrad. In undergrad, I really developed what was my work ethic. I treated this as my career. So I treated this as an investment in myself. I'm going to school. My school is being financed. I'm going to make sure I get the best of this experience. I took advantage of every opportunity that was offered to me in undergrad, every research experience, every chance for leadership, every chance for teaching experience. I was a TA for general chemistry and eventually became a TA for general micro at the end of my career. I also went to go the extra mile. So I took a chance for a lot of leadership experiences. As a leadership scholar, that was actually part of our scholarship requirements. You had to maintain being a leader on campus. You had to be involved and you had to make sure that you're mentoring other students. And then finally, just make sure you have a great support system. I started undergrad with my great friends and I ended them with my great friends, but I also brought new friends with us. And you're going to see them on the next slide. So here is my graduate school career. So this is where I did my graduate school here. This is the Medical Center in Little Rock, Arkansas, the University of Arkansas for Medical Sciences. My support system in graduate school was amazing. So this is the associate dean of graduate school at the time. Now she's one of the provosts for academic affairs at UAMS. This is my group of friends. Most of them are pictured here. This is my lovely mother, as you can see in this picture here. This is me receiving my white coat with two of my close friends who have now went on to become PhDs. And then this is us having fun at research conferences. So when you get a chance to do research in undergrad, get a chance to present it. Presenting research is fun because you get to build a network with people outside of your institution, and you get to just showcase your work. And these are people at Abercrombie that I became really great friends with, as you can see in the bottom picture. So in graduate school, I went to UAMS. I was offered to join part of an R25, which is a funding mechanism, which is meant to maximize diversity at this institution. I did 3 rotations. Each rotation, the first 2 rotations, I said, I'm going to be a microbiologist. I came from Dr. Patrick Sleiber's lab, and he is an internationally known microbiologist. I'm going to be a microbiologist. So I did a virology rotation, a malaria rotation, and then for the 3rd rotation, I said, let's do something that just seems cool, something I had never thought of. I did research in methamphetamine gene therapy, and that's where I ended up staying. So I joined the pharmacology department, and this is my mentor here, Dr. Eric Peterson. His research is developing new antibody-based treatments against methamphetamine abuse. In his lab, I learned the fundamentals of mentoring, and I learned how to manage a personal and professional life. Dr. Peterson is a great person, and I'm so lucky to have worked in his lab. He still helps me to this day, and I've graduated with my PhD. 2 years has passed. He's a great person. He's a great example of what to look for in a mentor. And this is where I fell in love with neuroscience, and that's how I got to where I am today. In Dr. Peterson's lab, I probably had another unique experience. So these unique experiences have really shaped my career so far to this day. I had a unique experience to design my own dissertation project. I had many research interests from my previous work, and I wanted to design a project that looked at the intersection of neuroscience, immunology, and drug addiction, and that's what I did. And that's how I got to my current research. So my current research I'm going to talk to you guys about is traumatic brain injury. And the reason that I went directly into traumatic brain injury instead of looking at different other aspects of neuroscience and drug addiction was my little sister. During my last year of my dissertation, my little sister was in a very bad car wreck and she experienced a traumatic brain injury. About a month later, she was back in the hospital. And these weren't even traumatic brain injury related, but they definitely had an association. She could not talk. She didn't know how to read or write. At this point, her traumatic brain injury from the different scans led the doctors to believe that she also had multiple sclerosis. And at this point, in a quick time, she had so many lesions, which is a hallmark characteristic of multiple sclerosis, that it had progressed in a very short amount of time. And so now she is back to functioning as much as a lot prior to her incident, but this initial traumatic brain injury is what stimulated my interest in continuing the research into neuroscience. So let's look at some of the annual statistics of traumatic brain injury. According to the CDC, there's approximately 2.9 million people that experience a traumatic brain injury annually. There's an estimated 13.5 million that live with a disability that is related to traumatic brain injury. And there's an estimated effect on the US economy of around $77 billion related to these 200 plus thousand hospitalizations that occur every year because of traumatic brain injury. So what is traumatic brain injury? It's a physical injury to the brain from an external force that causes impairment. This will lead to a lot of structural changes in the gross anatomy, and some of these alterations can be permanent. Currently, there are no clinical treatments for traumatic brain injury. The only methods that we have for treating traumatic brain injury currently are treating the symptoms. So traumatic brain injury is also a two-injury process. So here we have the primary injury. This is the actual external permanent brain damage. This is the head trauma. It can be a blunt impact, penetrating blast injury, a blast wave, such as a huge wave, or even just a strong acceleration or deceleration of the brain within the skull. The secondary injury mechanisms that's on the next slide are believed to be even more detrimental than the initial injury itself. These secondary injuries are characterized into numerous categories. They can be either looking at cerebral blood flow, oxygen, and energy consumption. So they can deal with brain edema, which you have an increase of fluid into the brain. They can be increase of, they can have ischemic events, which oxygen is not being directly transported into the brain, as well as glucose utilization is down because of different upregulation of transporters of glucose into this highly metabolic organ. And this is all because of different detrimental defects to the blood-brain barrier. So what is the blood-brain barrier? So while I was an undergrad, I had no idea what a blood-brain barrier was. But as I became in graduate school, the blood-brain barrier is what's going to be the biggest part of my research focus. So the blood-brain barrier is the structural components of the blood-brain barrier is blood vessels with large layers of specialized endothelial cells, astrocytes, and pericytes, and they're all being regulated by different neuronal transmitters. The blood-brain barrier has these endothelial cells, but these endothelial cells are different than endothelial cells that you'll find in your peripheral body. These have a unique ability to restrict the movement of different molecules into the brain, because the brain is theorized as this almost immune privileged event, but is also very critical to injury. So these BBBECs prevent the entry of toxins and other different microorganisms from reaching the brain. And these BBECs also they're able to do this restrictive permeability because of tight junction proteins. And these tight junction proteins are what I initially started to look at in graduate school, and they continued on into my postdoctoral career. So we wanted to model the BBB, but we wanted to do an in vitro model. So there's a lot of in vivo models using different model organisms, such as zebrafish, the rodent models, there's swine models as well. But we wanted an in vitro model because we wanted to use human-derived cells to study these different secondary mechanisms. But there are a lot of current models, but they didn't really fit the need of what we wanted. There's the single cell transwell model, and this is usually just using an endothelial cell. And these endothelial cells can be either from the brain microenvironment, or they can be from a peripheral derived counterpart. There's co-culture transwells as well. They use either these endothelial cells or the astrocytes. And astrocytes are a glial cell that are really important in maintaining homeostasis within the brain. There's the spheroid model, which uses different combinations of glial cells and is usually using some kind of extracellular matrix, which is an important part to be considered into a model looking at the blood-brain barrier. And there's also microfluidic models that start to incorporate the use of flow. And this cerebral flow is important in trying to recapitulate what we see in a person into these different cellular constructs. But we wanted to improve on these models. We wanted something that really looked at the dynamic interactions of cells in their environment. We thought as a team of engineers, so there's a team of engineers, there's a team of basic biologists, and there's a team of clinicians all thinking together what we need as a suitable in vitro model. We thought a suitable in vitro model of TBI would impose functional insults to the BBB, either they being mechanical, metabolic, or inflammatory, and also allows for the perfusion of blood and replication of the cellular biochemical environment. So this is really important because a lot of times in these BBB models, they're looking at the brain. So they are looking at cells of the brain, but they're not incorporating the blood flow or the blood component, which is a really important part of blood-brain barrier studies. So in our novel model, we created a modular dynamic model centered around a transwell. So we took this 2D model that has been highly sorted and used in numerous applications, and we created a platform that is centered around this transwell. It has sealed pressurized compartments that form around each membrane, creating distinct environments within the closed system, and even has a pump. This pump can impose wall shear stress, and this wall shear stress is important for regulating that flow of different blood and perfusion of blood and nutrients onto the blood-brain barrier. So we really wanted to have a model that can bring all of these sections together. And that's what we have here. So this is our model here. This is just a basic direction of blood flow. So you're seeing we have two parts with our sealed pressurized compartments separating the brain side and the blood side. So we're able to see blood flowing through. And then we have an electrode that is in both compartments. And this is for the measurement of tear. And as I'm going to talk about, tear is important for measuring the integrity or the ionic integrity of the blood-brain barrier. We use these different symbols here. So this is our LabVIEW software that our engineer engineered to measure tear. And we're able to take measurements from the blood and the brain sides. And these different measurements is also where our function generators are used to measure and acquire the tear measurements. And this shows you just at the bottom a fully assembled platform here with this as our platform without the top. And this is our actual arrangement. So we have these chambers that attach onto our transwells. Our transwells go into this bottom compartment, and this bottom compartment will serve as our brain compartment. So it's a sealed, pressurized compartment that we can also take measurements from and seeing what actual toxins that we're adding in are actually getting into the brain. And so for this, we had a couple of different benchmarking papers that we looked at. These benchmarking papers, and this is probably our favorite paper that we're looking here that we cited in our manuscript. These are some parts that we realized that we needed to incorporate and be considered into our model. So we wanted to make sure that we had a wall shear stress, that we wanted to look at the microenvironment, like having a basic basement membrane and extracellular matrix, which we got from our Matrigel. We assessed barrier function through tear and permeability. We looked at cell function and expression of different BBB markers, and we looked at co-culture. So we actually looked at a triculture. And in this triculture, we had our brain microvascular endothelial cells with our astrocytes and parasites. And this is what we use for the model of our blood-brain barrier. These are some of the most important cells of the blood-brain barrier. And so we wanted to have these incorporated into our model. So let's look at TEER. Let's go over a quick thing of what TEER is. TEER stands for trans-endothelial electrical resistance. This is the measurement of the electrical resistance across the cellular monolayer, and it's a reliable method to confirm the integrity of cell connections. For our cell constructs, we did seeding of endothelial cells on day 1, and then on day 3, we will start our experimental regimen. If we added different cells, these cells will usually be added on day 3 and 4, and then it will start on day 5. We usually have about 18 hours of wall shear stress with an addition of a biochemical insult and then a permeability assay. And this permeability assay will determine exactly what solute or what size of molecules are able to pass through our blood-brain barrier model. And so let's look at some of our data. So the first data that is shown here is our static condition. So let me see, where's my mouse? Our first models were using the HUVEC cell line, which is a peripheral cell line, and our human brain microvascular cells. We initially added those cells with co-culturing, and we saw we had an increase in TEER. So it was having an average of around 30 to 40 ohms to around 120. the addition of our parasites. So we aren't just a bioengineering lab. So we have parasites, but we also use these bone marrow derived mesenchymal stem cells. These bone marrow mesenchymal stem cells are very potent secretors of different mediators. So they also have very strong similarities with parasites. So we use them as our parasites for this model. And at the static condition, which means we're not turning on the flow of our pump, this is the tier we saw. We saw a dramatic increase from 120 ohms to around 450 being our average for our studies. Next, we wanted to look at wall shear stress. Wall shear stress is important in a lot of brain microvascular endothelial cell cellular physiology. We wanted to look at this to see, did we see a strong increase in tier as we've seen across multiple other papers? And that's what we saw. So here, just looking at our triculture, we had a very strong increase after our period of wall shear stress conditioning. And we believe this is because wall shear stress is changing some of the cell's cytoskeletal machinery and architecture. So it is different alterations that are occurring, tightening, most likely the tight junction connections is increasing the tier and increasing the, making sure that most molecules can't pass through our cellular construct. We also wanted to make sure that we looked at some parts that were important for TBI physiology. So this is a model for BBB, but we also wanted to make sure this was a model for TBI. And TBI involves a period of inflammation that's usually mediated or modulated through the inflammatory cytokine TNF alpha. TNF alpha is produced by activated immune and endothelial cells, as well as other members of the blood-brain barrier. And studies have shown that measurements of TNF alpha measures of TNF alpha have been shown in different in vivo models. So in an in vivo model, there's different types of injury. That can be a closed head injury model. That can be a CCI, which is a cortical constricted injury model. And all these models had one aspect of similarity, is that there was always an increase in TNF alpha. The sham group is the group of animals in these models that did not receive an injury. And as expected, there shouldn't be either detectable or high concentrations of TNF alpha in these models. So as you can see here, it's kind of overshadowed a little bit about our triculture group. But in our triculture group, which has had such a strong model of using those bone marrow derived MSCs, we start to see a decrease at 6 hours using our dose of 50 nanograms per mil of TNF alpha. We eventually repeated those studies, which is not shown here, using higher doses to get a dose response. And once we started moving up into those super physiological levels of TNF alpha, we started to see an eventual decrease in our tier being measured by our function generator software. We also wanted to have a great way to model ischemia. So, ischemia is one of those major characteristics that we looked for in our models in our secondary injury of TBI. So we looked at this model called the OGDR model. So OGD stands for oxygen glucose deprivation model. And as we said before, we know that during TBI, oxygen and glucose consumption is happening at a much slower rate than normal in a normal, healthy, metabolically active brain. This model is known to mimic the ischemia part. So it increases the permeability of the BBB, decreases tear, and it also can lead to the formation of brain edema. So for our model, we used a shorter time period for initial studies. So we did an OGD time period of 2 hours and a resuscitation or reperfusion time point of 4 hours. In a lot of the literature, you'll see that the OGD period is 4 hours and the resuscitation is 24, but these were just some of our initial studies. And this is what our initial studies yielded. So here we have our same groups as before. We have the HUVEC, which is the peripheral endothelial cell, our HBEC5I, which is our brain microvascular cells, our co-culture, which is HBEC5I and astrocytes, and then with our bone marrow derived mesenchymal stem cells serving as parasites as the triculture. So in this study, we look at the max tier. So the max tier was done right before the OGD insult occurred. The red is shown as our after OGD. So this is 2 hours of OGD insult. And then this is our recovery period. So this is at the last 4 hours of the event. And as you can see in each one of our groups, we saw a very sharp decrease, in some cases 50% in tier. And this is a great chance to show that our model can impose these insults. So this was just our initial characterization of our model. So we wanted to make sure that we could impose these insults and also actively measure them. So that was a major part. How can we measure these insults? Can we impose these insults? And what can this information lead us to? And now we're in our new phase of our experiment. So right now, I'm the person that goes around and collects volunteers to donate their blood and plasma for actually using human blood and plasma in our studies. Our studies are looking at the blood-brain barrier, but we must have the barrier part, which we seem to have in our NVU model. And we're also in the process of incorporating neurons and microglia, because we are a microglia, mesenchymal stem cell bioengineering lab. We're kind of an all-focus lab. And so now we're in our next part of understanding how blood incorporates into this model. How does this affect BBB physiology for understanding potential treatment options for TBI? And I just want to get to my acknowledgements, and my acknowledgements are my mentors. I have 5 mentors. So I have Dr. Cox and Dr. Wade, Dr. Gill. Max Skibber is our engineer. He has been a part of the bioengineering lab since he was an undergrad at Yale, and he is the one who helped create the platform. I've done all of the validation studies. We have Dr. Julia Livanov. She's another engineer who looks at a lot of blood coagulation factors. Dr. Supinder Bender, he's the person that looks at the different types of neuroinflammation. And all together, we're all part of the Center for Translational Injury Research. And now it's time for questions.
Hello, everyone. I hope we have some questions coming into Q&A. I have actually a logistics question for our team of whether Dr. Bolden will be able to see those questions as they come through. Oh, there they are. Can you see them down there, Dr. Bolden?
Let me see.
Do you see Q&A?
Yeah, I see Q&A.
Let's see.
Okay, I see them now.
Oh, now you see them. Okay, excellent. Well, I'm gonna bow out, and I'm gonna let you just answer these questions. Maybe you can reread them and then answer them. And then if other students have questions, please put them in the Q&A. Okay, thank you so much, Dr. Bolden. That was awesome.
Nope, thank you for the opportunity. Okay, so I have an anonymous question. So let me see. So what do you feel like there were any hardships that you went through doing undergraduate research? Yes, so initially I was just nervous. That was my biggest thing. I came from this rural area, and we were using these like this very sophisticated technology to do all of this research. And I was just nervous about it, just trying to not breaking anything. And I was also nervous about my time commitment. So at some point, it's during my sophomore year, that was probably my biggest year for just being involved on campus. And I was a RA, I was a TA, I was doing different events in the community, and I also had to make time for research. And Southern Miss, even though it's an undergrad institution, is a very strong research intensive facility. And so we would have parts of our lab courses where we would have to be doing lab experiments for an active class at 10 and 2 o'clock in the morning. I'll never forget, during our senior year, we took this class called Microbiology Physiology. We had to be there at 3 o'clock in the morning and then still have class the next day. So we'll be there from, each student would take different time points. Are you coming to do the time point at 1 a.m.? Well, I'll come do the time point at 3 a.m. and then you'll do the time point at 5 a.m. And you still have to do, if you're in a research lab, you still having to do your research in your lab. So there was a lot of hardships with that as well. So along the way, I did feature some doubts, and there were some hardships. So one of the hardships, the hardest hardships I had during my undergrad was me becoming sick. So I developed lymphoma during my junior year of undergrad, and that was the biggest part of my hardship. My grades suffered because I felt I wanted to be the same super person that I was before. So I wanted to continue to do as much as I was doing before. And one day I literally had to stop. I had to I've pulled back from doing a lot of different commitments because I was actively sick. And so I was still trying to maintain school. I was a hall director, so I was responsible for an entire residence hall at this point. And I just wanted to make sure I was fulfilling those main things I had signed on to do. So that was part of my hardest part, was like figuring if I can just continue on with doing everything and continue in school if I happen to go out. And hardships, I had plenty of hardships. So I was a first generation college student who parents, mom didn't really have money to send me to college. So all of my things I had bought for college were bought mostly through me and her working 2 or 3 jobs to help me out. So a lot of it was financial hardships as well. So trying to make sure I had money to actually attend. Luckily, I had a really great friends that for my first 3 years of undergrad, I rode with them to places, went home with them. Wherever there was a trip, I would contribute gas money or whatever. I really just had a great support system that would help me throughout this time. For the brain cells. So with brain cells, we've used initially to set up. So we had this immortalized microvascular endothelial cells that we use. Now our primary, our astrocytes and our pericytes are also, those are all primary. Those are actually received from different donors from research studies. And so we've switched to using a primary endothelial cell line as well. And I'm also looking into doing iPSC cells, so induced pluripotent stem cells. So that's an opportunity to take this stem cell, and because it has this degree of potency, you can change it to what cell type you need. And so I thought of increasing our tier by getting induced pluripotent induced pluripotent stem cells turned into brain microvascular endothelial cells, because it's shown in the literature you can get an average tier of 1,000 ohms just from using that alone. So that's what we've used. But initially we just started off with HUVEC and our HVECs, and then we grew into using co-culture and trying out using astrocyte conditioned media, trying to see if we saw differences in permeability and tier. And then we went on into using our bone marrow derived mesenchymal stem cells. What I enjoyed about microbiology. What I enjoyed about microbiology was really a lot of just the protein aspects of microbiology. So looking at microbiology earlier on was just when I was in high school, it was more looking at just doing biochemical testing, looking at if we're getting bacteria, doing basic gram staining, doing catalase tests, trying to figure out the identity of the bacteria. So looking at these different aspects of just trying to determine what's the identity or the classification of the bacteria. And at that point, I didn't even realize that I was pushing myself to microbiology, but I really thought it was just biochemistry. So I also wanted to know about the different virulence factors and how are they encoded into these different organisms. And that's where I got involved with Dr. Sleiber's lab, looking at doing amplification of genes, trying to figure out and profile and characterize these different staph strains. There were ones that were causing infections in atopic dermatitis patients, which is a rash that people get and usually have a lot of social anxiety about, and how these different virulence factors they were producing in these different environments were allowing them to cause infections to different degrees of severity. How much comfort? When I describing my conclusions or discoveries made in my research, it's just something that came from doing that. I had done as much work as I could with my research. And so working with my mentors, they were always the sounding table. And I asked them to be as honest with me as possible. If you thought this was a good job, tell me. If you think I can improve on it, tell me. And for this validation model, this has probably been the most amount of back and forth with a mentor over a project besides my dissertation project, because this is something that we're hoping is going to revolutionize the field of using in vitro models to study different neurological disorders. And it's just something that really does improve with experience. There are some aspects where you're working on things and you're just like, I don't think something is going to go well. And that happened not recently this week. So there are certain parts I've done some of these experiments repeatedly. Like, I should know how to do this experiment, or I should know that this is setting up. And every now and then there's either something that's a small platform issue, or there's something with the cells, or we recently had the worst of luck. Everyone usually placed their new cells on Friday if we're plating new cells. And then over the weekend, someone might come and check in them on Sunday, but usually no one checks on them until like 5 o'clock or 6 o'clock Monday. So we're at a clinical lab, so we go with a clinical schedule. So most people are here in the lab at 5 and 6 o'clock on Mondays. And at Mondays, on Monday morning, I came in to look at, just check on everyone's cells. And the media was a completely different composition and color than we had saw before. Something had happened with the incubator, and all of our cells were dead. We had 6 incubators worth of cells, and some of these cells were stem cells from donors. Like, these are some stem cells from donors of people who had just given birth, like umbilical cord stem cells. These were stem cells that people had reprogrammed for different studies or active studies that had been going on, and they were all dead. So it was just a really big setback. And we had an issue with this same incubators doing that again, maybe like 6 months later. So there's always going to be a setback, or there's always going to be something that's going to come in, but experience will teach you that it's going to be okay, and that you can always figure out a way to just grow and learn from it. And that's what I did. I think being a biomedical research student is just learning. And me starting early as I could, starting with just doing like wildlife biology and ecology. It started with field sampling to doing biochemical testing to running full experiments. And so it was just something that you learn with as time goes on. In grad school, you mentioned rotations. What is a rotation? How does it work? So rotations in graduate school are an opportunity to get a snippet of research of a particular research lab. So in my graduate school back then, our research schedules have changed. During your first semester of graduate school, you're taking courses and you're doing research, and you're doing research rotation. So a rotation gives you anywhere from a month to 8 weeks to see if you really like working with this person as your mentor. And during grad school, when you're doing like a PhD program, you get to work with, you usually choose 2 or 3, and if you figure out at the end of your career, at the end of that first semester, that you still don't like your rotations, you can do another one. And so I did the first one with a virology lab. So she was a new professor, and I had never worked with cells before, not human cells. I had worked doing bacterial culture, and bacterial culture and human cell culture, or cell culture in general, are completely different. And so I did that research in rotation. And it's funny enough, I'm doing cell cultures now, but during that time, I said, I would never do cell culture. I was adamant. I was like, after this, I mean, I had a great experience in the lab, and she's a great mentor, but I was like, I'm never going to do cell culture. And that's exactly what I'm doing now. My second rotation was in malaria immunology, but it was more protein work. I was studying how to stimulate immune responses using these different protein kinetics of malaria, and trying to figure out if a way we could eventually prevent infection of malaria. So I was working in this second lab. My third rotation I took because from my mentor I had put on start in graduate school, we get a peer mentor from a higher level graduate student. Sometimes they say you do it 3 different ways. The first way is your last rotation is who you really want to work with, or who you don't think you're going to be interested in. And so I chose my last rotation as someone that I thought, I'm not going to want to do this. I am going to just get in this lab and learn the experience, and it's going to be just a great way to learn about different things of biomedical science. My third rotation is where I ended up staying. My third rotation is where I fell in love with having another great mentor. I had a great mentor with Dr. Bell and Dr. Sleiber Pat and Dr. Shu in undergrad, and I really wanted someone that I felt like that was similar to him. And he gave me a unique opportunity that I couldn't pass up. Most of the times when you're starting your grad school after you're done your rotation, and you usually have like a really small research project that you can do in like a month or two during the rotation. He gave me a project that was part of his major grant. So his major grant submission, he gave me a part to work on. And during this time, I realized I really like what I was doing, but I also could develop my own research project, and I couldn't pass that up. So I was like, I'm going to join his lab. And he was like, I don't have experience really in immunology. He works with developing antibodies, but this is more pharmacological. This is like pharmaceutical science. You're developing a product. You're not actually doing immunologic testing. And so at this point, I was set and I chose him. So rotation is just a great way to get an experience of what you want to do. And being in an interdisciplinary biomedical program is a great advantage because you're not confined to one department. Like there's of course different departments. While I was at UAMS, there was like pharmacology, microbiology, immunology, biochemistry, genetics, cell biology. But when you're in like an interdisciplinary biomedical science program, you can do research rotations with any mentor that has an available slot. And that's what I did. So it was a great chance to just see what I wanted to do. And I ended up working in methamphetamine gene therapy and also telling people that you're working on treating methamphetamine. They always ask you, how much do you know about it? So there was a kind of a cool wow factor I had in grad school as well, just telling people. My mentor, he was a very funny guy. He was the professor that while everyone else was maybe being serious at a meeting or like an auditorium meeting, he was the guy in the back, red in the face, laughing. So he would always tell people, oh, when they ask you what you do, just tell me you work in a meth lab. So it was a great time working with him. And he's been a great mentor in and outside of the lab. I first got started with research. So it's kind of hard to say. So it can either be my first research experience in high school or my first research experience in college. If we look at it in college, it was because he he basically told me I was joining his lab. Like literally, I walked on the floor. I was looking for because I took classes this summer, and in the summer when you come in as a freshman, you just take core courses. Now it was time for an actual advisement, and when you come in the fall, you meet with you have to figure out who your advisor is. And I'm wondering upon to the we had 6 science buildings, and the biggest science building where a lot of the classes were taught was Johnson Science Tower. It's a 13-story building, and I'm going to the 11th floor that I don't even know if I have access to find this person. And he found me first, and so he basically told me, "Oh, you're joining my lab." He had started off in Mississippi. He had went to do his PhD at Yale. He had did postdocs at Harvard and NYU, and he had back to Mississippi. So he was a person that was really invested in having other African American students like himself working with, because he was the only African American person in our College of Science and Technology, and so he wanted me to join his lab. So he brought me in. Now in high school, I was pretty proactive. I didn't even know what I was signing up for in the aspect of doing research, especially going to Hattiesburg on the weekends with them having the Natchez has a transit bus, and the transit bus was paid for through research dollars to bring us there on the weekend. And we will stay in this basically they had these extra dorms they were used for visitors and conference events. We would stay there on the weekend, then go back, stay there on the weekend. So we'll leave on Friday right after school, get to Hattiesburg about 5 or 6 on Friday, go do our research, head on back on Sunday afternoon, and we would do that. So my weekends were always booked. I was doing research, I was working after school, and doing the tutoring program at the same time. But my really research experience, I guess you could say, started then. And so I was doing many projects on the weekend. Usually it was just collecting field samples and collecting water samples and doing like colony forming unit testing and stuff like that. But that was my first research experience, was in high school. And then my first, I guess you can say, biomedical research experience was really my first year of undergrad. Within my first day of going to advisement, he told me later on that day, I'm going to all the welcome week activities. He was like, oh, don't forget we have a lab meeting this evening. And so I went to a lab meeting and I started from there. Yes, there are research programs available for students to do without research experience. That's what the research experience for RU programs or the RU programs. And if you're ever looking for some, I'm always in contact with people because I've been a recruiter for programs for that. So if you ever need to get in contact with information about one, you can always get my information from Dr. Morano. But these are great opportunities for people that don't have research experience. A lot of times at different undergrad institutions, we don't have an opportunity to get into a research lab because those spaces are highly coveted, and we don't have an opportunity to get into that actual environment. These give you the chance to go somewhere for the summer and pay you pretty well. So this was years ago, 2011. Um, it's been 10 years from now. My first RU was paying me $5,000 for the summer. It gave me $750 for meals, which I also could use my stipend money, and they put me in an apartment for the summer, a fully furnished apartment. And so this is a great chance to go to a different city or a different environment, gain research experience, and also get you a really great outside mentor. Because this can be somebody, he or she could be someone that eventually down the road is your letter of recommendation, or when you're done with undergrad, they could also be the person that you work with. I would have joined Pat's lab, but he was retiring from taking in active students. He was already a senior professor. He had been internationally known. He was in the CDC record vote for his accomplishments with toxic shock syndrome. So he wasn't taking any more students. He already had taken his last graduate student while I was doing his research rotations. But if he was, I would have definitely went with him because I had such a great experience working with him. So if you're ever looking for research experience, first, while you're at your institution, ask your faculty. They usually have their own active research programs going on, or know of someone in the area, because you're in Houston. There's always some kind of position, especially with maybe not as much now because of the COVID implications with people coming on into their campuses, but there's always someone that's looking for an undergrad research assistant, someone to just gain experience. So you can even gain experience just in Houston alone, because there's so many institutions, and the medical center is right up the road. There is so many opportunities for summer research summer research or school throughout the school year research as well. So just be an advocate. If you need to talk to me or talk to any of your faculty members, just do that. I think that'll be a great experience. And it's also good to know if you're interested in it before you decide to do a full research-based degree, such as the PhD, because if you don't know if you like it, you don't want to commit to something and you're just, I'm not sure if this is going to be for me. But joining early on with doing these event of research experience, like, oh, I can do this, or I can learn, this can teach me a lot. So I would just say get that experience as early as possible. And even if just doing something on Fridays or working in the evenings, if you have time, you can find someone that is very that can accommodate your schedule. So no, so our BBB model has not been used outside of the country. The reason being is because this is our mentor also has his own company, his own biotech company, and it is licensed through the biotech company and University UT Health. So right now it is only even the full mechanistic drawings are only able to be shown in our lab group until we get a complete patent on this project. But right now we are publishing the data and getting the information out, and it's the same data that you guys saw on these slides. So this is publication grade data that's being shown here is what we presented out in this PowerPoint here. But no, not right now. It's not being used outside of the country. It is only used in our lab group. So it is only found here in Houston. Yes, so we are testing specific hypotheses. Some of them I can't say because we're submitting a grant on, so those are some of my own intellectual ideas, but we are looking at the ability of blood into our model. So on the back of the model, which you really couldn't see in the figure, there's a reservoir. That reservoir is where our cell culture media is. And so that cell culture media is what passes on top of our cells, which represents the blood side of the blood-brain barrier. What we're going to do is take out media and eventually put in whole fresh blood. So we've already been using human plasma, but we haven't started incorporating blood into it. So we want to see what the perfusion of blood has on affecting cells of the blood-brain barrier. And as more realistic as we get to actually human, like in vivo, like inside of a whole person's parameters, we can eventually look at testing out different therapeutics to be eventually screened to go into clinical trials to help hopefully one day provide better medications for TBI patients. Because as I said before, a lot of the TBI medications now only treat the symptoms. They do not treat the mechanisms of the disorder. And so they're only treating with like either decreasing blood pressure to certain degrees or decreasing the intracranial pressure, but they're not stopping neuroinflammation. And neuroinflammation is probably one of the most detrimental parts of TBI. This TBI initiated inflammation causes tissue death. And this, as we know, your brain tissue is highly vital for your cognitive abilities. So if we can figure out a way to prevent this tissue death, so as soon as someone comes into the ER, we can give them this therapeutic, and this will hopefully prevent some of those detrimental effects from occurring. I wanted to see any difficulty staying focused on your research. Yes, there are certain times staying focused on your research can present themselves as like a little issue, because there's always going to be these new avenues, and I'm one of those people that always looks at, okay, this research has this implication that can go here and there and over here. And so it's like, no, you need to make sure that you're still staying focused on your main objective. I think one of the biggest things is making sure that I understood and established what was my goal. What is my research goal? Why am I doing this? And until I get that goal, or at least a sub part of that goal completed, that's when I can start looking at the other aspects of it. I felt like it was too much when I was sick, or wasn't being true to myself about what I could do at that point. You also have to be very, when you're doing your research and joining research programs, be very well aware of yourself, knowing what are your other commitments you have, and make sure they don't overlap, because those can become conflicts of interest. Your time is being diverted to other areas, and it's not being focused on what you signing on to do. So there can always be a lot of personal issues that can inherently distract you from your research. For me, as I said, my mom was my only support system, like financially, even in grad school. So I was working, getting paid a stipend, but we were helping each other. And so I easily got distracted when her job shut down. This is before the pandemic, where her job was sold, so she lost her main sources of income. So I became distracted at this point because I wanted to look at other sources of income to help supplement her and help her with her different issues that she was dealing with. If you wanted a career in a biomedical lab, what kind of degrees or qualifications would you need? So that's the cool part about being a biomedical lab. You don't have to have any degree to work in a biomedical lab. You can just start getting your experience. Like, we have high school students that are working with us now. Like, these are sophomores in high school who have contacted my mentor, and they have said they want to work with him. And so he knows that I want to have an undergraduate lab. So they usually end up working with me, but you don't have to have any kind of degree or qualification for a biomedical lab. If you want to work in a clinical lab, you have to have either like a medical laboratory science license and certification. But for biomedical research lab, you just have to want to get involved in finding a lab to take you on and get your experience. Of course, when you go into graduate school, having a bachelor's, usually in a life science such as chemistry or biology will help, but you can also be focused in different areas and get accepted, as long as that you can prove or demonstrate that you have experience and understand a lot about those concepts of what of the career that you're trying to go into. I know some students that did our in the PhD program with me, their bachelors were in English. They took biology, they took organic chemistry and physics, but they were actually English degrees. And having those other degrees, even for those who want to go into medical school, but taking those basic biological science courses can be a lot, because learning how to read and write and communicate your information effectively will help you stand out among the other applicants. I commend you for stating how you work with one career choice and ended up with your passion. What advice is to have a strong support system? Follow that true passion. Okay, so this is a great one. So not having a strong support system, I think this is where you can start to build one. So I had a very strong support system with my friends. We were determined to succeed, and we some of them, like I said, we came in together. So there was, I'm trying to remember, count all my friends that came in together. There were 6 of us that came in together from my high school. Most of the people from my high school went on to other colleges, but we went to Southern Miss. 6 of us came in together, and we met other people, and now we have a group of about 17 of us that we all met from undergrad. And it's just, we have to support each other. So you can start to look for other people to build your support system. And also your support system can be your advisor. Dr. Bell, who was my advisor that I talked about early on, he has been my support system from the beginning of undergrad to me being in my going into my second year of a postdoc. He is whenever I have an issue with anything, like I'm trying to write a paper, I want to apply for an award, I need someone to tell me it's going to be okay and that I can eventually get through this. He has been that as a mentor. So your support system can even be sometimes the faculty or the or your advisor. I'm not sure how it's set up at UHD. I'm not sure if you have like a faculty advisor or you have a someone in an advising office, but it can literally be the people that you are meeting with. And that's sometimes what a lot of students have to do. I was lucky that I had a group of people going to college with me. We met one of our closest friends in our group. He was the only person from his hometown that went to Southern Miss, and he didn't know anybody else at Southern Miss. And so me being, I'm more introverted, um I have more introvert tendencies, even though I've done a lot of leadership experiences. I saw him in one of my classes, and we had been in classes for a while. And one day he just asked for notes, and then one day he saw me with my friends, and he thought that we looked approachable. He said, "Hey, can I study with you guys for the second exam?" We had took the first exam, and my group had done pretty well, and so he was like, "Let's join together and study." That studying went on from being just meeting to studying to like, you want to go to McDonald's or you want to go get something to eat, or let's go to the cafeteria. Let's all wait till you get out of class and we all taking classes together. So you can build your support system. And following your path, and but you're trying to follow your passion, don't let anything stop you. If you need to, whatever you need to do to get there, do it. And that means if you have to do like extra office hours, because there are going to be some parts of some courses that are just going to be difficult. Organic chemistry, organic chemistry 2, difficult. I had to do extra studying hours, office hours with my professors, looking at tutoring, contacting professors, asking them about activities for research experiences, getting to know faculty, knowing deans, so I can have these strong letters of recommendation and getting involved. Because believe me, getting involved and having not only just succeeding in class, but having that person that's showing that you know people at your institution will make a big difference. Showing that you can have letters of recommendations that aren't just saying, "Oh, you were in my class, you got an A in my class or a B or whatever," and that's it. But having that person that's saying, "Oh, they worked in my lab," or "They always come to my office hours, they're always answering questions," or they're trying to volunteer, they can really speak on your character. That makes a big difference. Like who you like, showing that people actually know you and that you're committed to your work, you can get there. And you can always find outside mentors as too. I serve as mentors to students that are not at my institution, because at UT Health we don't have undergrads, and so I love undergrads because I teach undergrads now. And so I'm always like, if you need a mentor or you need a support system, just to, like, if you're having a moment to go through, contact me or contact other faculty. There are different people that you can help build your own support system with. So I was off set to start graduate school and had just received a few acceptances. Unfortunately, I got sick from traumatic brain injury and went through a similar experience to your sister. While you were going through the phone, what kept you driven and motivated? I think the biggest thing that kept me motivated was that um I had started this and I wanted to finish it. There was a lot of times I went to counseling during this time as too, because I started to suffer from depression, and and it just really brought me down. I think I felt so self-defeated at a lot of aspects that it would start affecting my performance in classes, because I went from being almost all A student and double majoring to I'm just trying to get by, and it caught the flags of some faculty, because some faculty that I hadn't even interacted with before, I guess I had a reputation for being a pretty decent student, and so they would pull me aside like, "What's going on? Do you need someone to talk to, because you're not performing like you normally do." And I think it's just making sure that you know what your goals are and you're not overly stressing yourself out. You can get there. What really also kept me motivated was my best friend. So in my group, there was me and my best friend. We have been best friends since preschool. we have been literally best friends since we were 3 and 4 years old, and we're still best friends to this day. And then when I was sick and needed someone to pick me up, she was there and has always supported me. I emailed her right before this presentation, letting her know that I'm about to start talking about my research and my career trajectory. And she sent me, I think, over 20 GIFs and a whole paragraph saying, like, a gift saying, like, this is what you're going to do. You're going to do great, and talk to people and just get it out there. She's always been my support system. And I've been blessed to have someone like that as my support system. So I had a really great, as I said, I harp on that so many times, mentorship and support system was what really made my whole career. See. Okay, so the next one says, what skills are needed to do in order to do research? In order to do research, commitment and determination. First, before you get into any subspecialty or subdiscipline, commitment and determination, because there are going to be a lot of times when an experiment that you have done 20 times to perfection is going to fail. And it's going to be because of something simple that you missed out on or didn't pay attention to. It's going to be a lot of times where you're going to do a new experiment and it is going to just fail directly off the board. And so you just have to be committed to what you want to do and determined. Also, being able to take criticism. In the research community, there's always a lot of scrutiny and criticism about different projects and stuff that is going on. And so you want to make sure that you develop a tough layer of skin, but also maintain your own self-respect as well. So while you're doing your research, always get input. Don't feel like you can't ask anyone a question, especially when you're first starting off. There are other people usually in the lab or either just your mentor that you can go to. So I think the first part is having an inquisitive mind, being able to be determined, and then just also just being committed to your work. And you'll be surprised how far it will take you. Down the road, I really see myself being an undergrad mentor, like an undergrad doing an undergrad research lab somewhere like a UHD. But I also see myself running a biomedical research program. So not doing a biomedical research training program. So like Lewis Stokes, that you guys shared, like the LSAMP program, I want to be a part of that program where we're making sure that we're maximizing the success of all of our students. So I want to make sure I'm a part of that, training the next generation of scientists or just biomedical students. And so I see myself leading and directing that type of program. I want I want the students of every background to come in and feel like that they can succeed. That's what I want to do, because I believe, you hear people say I have natural talent, and there is a lot of natural talent, but talent is something that is created. And we have the opportunity, we have more resources now than ever before to create talent, and we can create talent in and outside of the lab. And so I want to be creating talent inside the lab, doing the research experiments and helping each student create their own hypothesis. But I also want to be able to help them reach their goals. So even though I have my goals as a researcher that I need to have answers. So I want you to be able to help me answer, looking at this hypothesis, looking at this receptor on this different type of cell that's found in the brain. Well, I also want to know, how can I help you get to medical school? How am I going to help you get to graduate school? If you want to go on and get your master's of public health, what can we do along the way to make sure that when you go from this one institution, this one area, you are succeeding in every step along your pipeline? And that's what I want to do in those graduate programs. I want to change career outcomes for different students across different areas. So there weren't really any ethical areas, dilemmas at this moment, faced with in our experiment, because we've only been using certain donors from campus, and they're just for their own, just for their plasma. Now the other cell types that we received, there's all been patient consented, or people have been enrolled in a research study with a whole IRB, and I've been added onto that IRB, so that we have made sure that we don't have any kind of ethical dilemmas at the moment. We haven't taken like any other type of stem cells besides like umbilical cord or like bone marrow from bone marrow people that come in to donate. But we don't have any ethical dilemmas at the moment. Now, in some other aspects of our lab, because we are a full clinical lab, those could present themselves, but we don't really have those in my area, in my research project at the moment. And this final question: in regeneration, like how crustaceans replace a broken limb of the brain is still under research, or is it? No. So our lab is part of a program, and our program is known as the Program in Regenerative Medicine. So we are a lab. As I said, each time I try to say what we are as a lab, we have multiple groups in our lab. So it's different areas, but one of our biggest areas is the Program in Regenerative Medicine. So we're using a lot of stem cell therapies, stem cell based techniques to treat pediatric traumatic brain injury. So one of our lab areas is pediatrics as well. And so we are actively carrying out different studies to use for the treatment of pediatric TBI as well as adult TBI. And we have some that are in clinical trials now. So the program, I think regenerative medicine is just now starting to even get a bigger push. And there's a lot of different trials going on nationwide and across different countries in regenerative medicine. I think it's just, it's starting to hit an area where we can see a lot of new improvements. And I believe that is all of the questions. But I just want to thank all of you for attending this seminar. And if you have any kind of information or any kind of questions, you are welcome to get my information from Dr. Morano. You can contact me, even if that's just like, oh, I'm interested in taking this kind of course, or I want to find a research experience, or I want to have an outside mentor. Yeah, just contact me. Anything you can, whatever you think of, you just have a straight scientific question or needing help about moving in your next phase of your career, just let me know. And I think that's it.